Perfect. So welcome, everyone. Thank you for the introduction, John. I'm going to present indeed a preclinical case study analyzed with the Monolix suite that aims at predicting optimal scheduling of drug combinations using a population PK/PD model in lung cancer xenografts in mice. And I will show that the building of a tumor growth model to perform these predictions is now very easy to do in Monolix, thanks to the new library of tumor growth models that has been released last year. First, a brief introduction of our own case study. It is based on some real tumor growth data published by Imms et al. And modeled in this paper by Schneider et al. The model I will present in this example is quite similar, but not exactly the same as the one in the paper, because I have built it on the new library of models in Monolix, which provides slightly different models for tumor growth. So this research studies the effect of bevacizumab, a treatment employed in combination with pemetrexed and cisplatin, which is the first-line chemotherapy for advanced non-squamous non-small cell lung cancer. And bevacizumab is given in combination with chemotherapy because it potentiates its cytotoxicity by inducing transient tumor vasculature normalization. This activation is not instantaneous, but it typically occurs within a period of a few days after administration. And therefore, it is more efficient to administer bevacizumab a few days before chemotherapy. So the goal of this study was to estimate the optimal gap between administration of bevacizumab and chemotherapy to reach full cytotoxicity activation. Here is the workflow that I will follow to analyze this data and answer this question. I will show the main steps of the modeling and simulation workflow performed with the Monolix suite. So starting with data exploration or the longitudinal data that we have from 77 xenografts in mice with different dosing schedules. Then I will use this data to build a population model in Monolix with a stepwise development of a tumor growth inhibition model. And finally, I will use this model in Simulx to predict the optimal time gap for cytotoxicity activation. I will show most of these steps directly on the slides, but also a few of them directly in the Monolix suite. So first, indeed, I will start with showing you the data set directly in Monolix. So it will be a bit more interactive. So here we see the tumor growth data measured by fluorescence in lung cancer xenografts in mice. So we have the longitudinal data split in 5 groups. And the group corresponds to mice that have received a different treatment with about 15 individuals in each group. The time here on the x-axis is in days. And the dosing times are shown as dashed vertical bars. So on the right, you have the control group with no treatment. The yellow group here has received only chemotherapy and no bevacizumab with 3 successive doses with 14 days between each dose, so 2 weeks of interval. The group in black here on the left has also really received 3 doses of bevacizumab at the same time as chemotherapy. So for each vertical dashed bar, we have the 3 molecules that have been given together. And finally, the 2 last groups have received also 3 doses of bevacizumab, 3 days before chemotherapy for the second group in pink, and 8 days before chemotherapy for the blue group. So the goal of the modeling workflow will be to build a model to capture all this data from the different groups and decide which dosing schedule gives the best result. Estimating a model on this data is especially interesting because the treatment effect seems quite small here. So at first view, it is difficult to see which group corresponds to the strongest effect. By adding a mean and standard deviation curve, like this on the plot, we can, however, notice a global small growth decrease after each dose, especially in the 2 groups on the left that have received bevacizumab concomitantly or 3 days before chemotherapy. Now, if I focus on the group receiving only chemotherapy, so this group here with a log scale, we can also notice that the effect of chemotherapy itself shows a delay of a few days after administration. And now, if I have a look at the control group, which is here, so instead of capturing directly the treatment effect, it will be easier to start the modeling workflow with a first step where we model only the control group to find an appropriate tumor growth model. So from the shape of the data for the control group, we can say that the unperturbed tumor growth, for example, we can see for this individual 1, the tumor growth is fast in the first days and then decreases a little bit with time without exhibiting a clear saturation in the during the duration of the observation. So this characteristic noted during data exploration will help us to choose a good model for the tumor growth and combination therapy. So I will now show on the slide how we build this model in Monolix. As I have mentioned, the easiest is to build a model in several steps rather than directly on the whole data set. And since Monolix can filter part of the data set for the analysis, it is very easy to first select the control group to estimate a good tumor growth model on the unperturbed tumor growth, then complete this model with its results, so with the last estimate, with an inhibition effect estimated on the chemotherapy group. And finally, complete this model to take into account also the combination of bevacizumab and chemotherapy. Thus, at the end, I will obtain a powerful model estimated on all data. So I will start with the first step. To model the tumor growth, I can rely on the tumor growth library in Monolix available in the tab Structural Model. This library contains a wide collection of tumor growth models based on the list of growth functions shown on this figure here, taken from the online documentation of the library, with typical predictions for each function in log scale here. In the library, the functions are organized in functions without saturation of the growth or with saturation. So functions that predict a maximal tumor size, which is the carrying capacity. So here's a list of such functions is given here because saturation is selected, and I can select any function to model my data. In addition, this is a modular library, so I can select a few other choices to customize the model, such as whether the initial tumor size should be estimated as a parameter or directly read from the data as a regressor, and whether I want to include additional features in the model, such as angiogenesis or some treatment effect. But for the first step, I just need a simple growth function in the model without treatment or additional feature. So with this library, I can very easily test different hypotheses on the tumor growth kinetics by selecting some function that looks like it could capture the data. So here, I would not select the exponential growth function in purple, where the growth rate is constant, because I have seen in the data that it would not be adequate. But maybe the linear growth function in pink, or the exponential linear in red here, which begins with an exponential phase before switching to a linear phase. And I could also try growth functions with saturation also, that could capture the decreasing growth rate as the start of the saturation phase. But in this case, the parameter characterizing the carrying capacity, so the maximal tumor size, might be difficult to estimate since we do not have enough data to see clearly the saturation. In addition, after trying different growth functions on my data, I can quite conveniently compare the models in Sycomore, the dedicated application of the Monolix suite for this, to compare for all the different models that I can try on the data, the objective function in each case on this table, and also the individual fits. So for this case study, I have actually tested all the available functions on my data, and I have sorted the results here by big. So the best models are shown on the top. And the best model is the exponential linear model with a sharp switch between the exponential and the linear growth phases, as we can see here on log scale on some of the individuals. And this gives the best result. Other functions, such as the core growth function, characterized by a smoother transition between the exponential and linear growth phases, capture also quite well the data, but a bit less well. And as a comment, the Simeoni function that you can also see here as the second model is actually equivalent to the exponential linear function, but it is implemented with an ODE. So it can be combined easily with a treatment effect, while the exponential linear model is implemented with an analytical solution, and it cannot be combined in a more complex model. So since I would like to extend my model now with the treatment effect, the Simeoni growth function, which is equivalent to the exponential linear, is the one that I will use as a starting point for the next step to capture the data from the chemotherapy group. So for the next step, I select the Simeoni growth function in the tumor growth library. And now, instead of no treatment, I choose in the library a model where the treatment is described with a PK model that directly reads the dosing information from the data and predicts a drug concentration that affects the tumor growth. And then I have to choose in this menu the options I want for the tumor growth inhibition. So in these different options, there are two possible killing hypotheses. The log kill hypothesis, where exposure to a given amount of treatment kills a constant fraction of the total tumor cell population. And the Norton-Simon hypothesis, where the killing term is proportional to the growth rate. For the killing term, different linear or nonlinear dynamics are available that are represented here. And the model can also take into account emergence of resistance to the treatment, which does not seem to be the case here, so I would select none, or delay of the treatment effect, which we have seen in the data with two possible options here. So all these choices are, of course, documented on the Monolix website with clear explanations of their differences, the corresponding equations, and figures of typical predictions to help choose an adequate model. But when the treatment effect is small, such as in this example, it is anyway difficult to distinguish in the data a shape that would be characteristic of a particular dynamics. Of course, prior knowledge on the biological mechanisms could help. But as we have seen, if prior knowledge is not available, it is also quite easy to test different hypotheses by selecting different models from the library. And so in this example, I have simply tried all the 16 possible combinations to find the best one. So here is the model corresponding to the best results that I have found. And this is the model as written in the library. It is composed of the Simeoni tumor growth function in this ODE with a Norton-Simon linear killing term defined in the variable k and applied proportionally to the tumor growth, proportionally to the growth rate with the variable k3. And the k and k3 are linked with a signal distribution module here in yellow that induces a delay in the killing signal. So actually, to model my data, I could not apply directly this model that is in the library because the treatment exposure is modeled with a default one compartment PK model, which is defined implicitly here with the function PK model and these two parameters that correspond to the V, the volume in the compartment, and K, the linear elimination rate that have to be estimated in Monolix. However, in this case, we have no PK data to estimate the parameters, and this PK model is actually not the most appropriate. So in such a case, it is very simple to use the model from the library as a basis to edit and adapt the PK part, for example, here. And this is what I have done. So I have replaced the default PK model with a combination of two PK models written here, proposed in the publication where this data was modeled. And we have one PK model for cisplatin and one for pemetrexed, with PK parameters that are fixed to literature values. Now the treatment exposure defined here is the sum of the two drug concentrations for cisplatin and pemetrexed. So this is the effect of chemotherapy. So this, in the end, is the best model to capture the data in the chemotherapy group. Now the next step is to include to extend again the model to include the effect of bevacizumab in order to be able to model all the data at the same time. So again, for this, I have used the extension proposed in the paper with a third PK model for bevacizumab and an activation term for the killing signal, displayed here in orange, that depends on the bevacizumab concentration, Cbev, and a parameter delta. And in addition, we have seen that there is a delay in the activation of the chemotherapy effect by bevacizumab. This delay in activation is actually represented by a lag time with a parameter T lag here. This lag time is in the absorption of bevacizumab. So it delays the concentration of bevacizumab, and thus it also delays the effect for the activation. So this is my final model, and here are the results I get with this model. The data is not rich enough to estimate well the inter-individual variability on all the 7 parameters, but I have still kept 4 random effects, and we can see their standard deviations here, with some correlations between the random effects that we can see also here. So all the parameters are well estimated, as you can see here, with small standard errors. And on this figure, you can see predictions based on this model for each group, displayed as percentiles bands and overlaid with the data from each group. And we can see that the model captures quite well the data in each group. And we notice that the treatment effect seems a bit stronger in the group that has received the bevacizumab 3 days before chemotherapy. So the pink group here, we can see this a little bit in the predictions. And to check whether this is really the case, I can now use this model to predict the treatment effect with different time gaps, different than in this initial data, to check different time gap between the administration of bevacizumab and chemotherapy. And I will do this in Simulx, the application of the Monolix suite for simulations, with the goal of finding the optimal delay between bevacizumab administration and chemotherapy administration to have the treatment effect. So now I will show directly in Simulx how to plan and perform my simulations. So here is my Simulx project. I will actually increase a little bit the size. No, it's okay. Yeah. So I will let it like this. I hope that you can see the different parts of the interface. So this is a Simulx project that I have obtained by just exporting my final model estimated in Monolix to Simulx. And now Simulx has automatically defined in the tab definition here all the elements that I need to resimulate the model as in Monolix, such as the model that we can see here, that I won't take the time to detail, all the population parameters estimated in Monolix that we can see here as a vector of values, or in treatment, we have also the dosing information from the original data set that I won't take the time to detail either. But so based on all these elements or on new elements that I can define, I can perform in Simulx 2 types of simulations in the tab Exploration and Simulation. And I will start by using the tab Exploration. Here, I can simulate a single individual based on typical parameter values, and I can interactively change the parameter values and the treatment characteristics here. By default, the simulation uses typical parameters the fixed effects estimated in Monolix that are stored in this element that is a vector of values. And this, I can also see the corresponding values here for the parameters. As default, it also uses as output in this element tumor size variable with a default time grid that you can see here. And as treatment information, I have here the tables corresponding to the individual doses, for each molecule. So this corresponds to a table of individual doses for bevacizumab. This is for pemetrexed, and this is for cisplatin. And with this slider here, I can select the index of the row in this table that I want to read. For example, if I select 50, like this, This changes a bit the prediction because it corresponds to an individual in the group that has received concomitantly chemotherapy, so 3 doses of chemotherapy and 3 doses of bevacizumab, while the individual one is from the control group, so there is actually no dose that is given. So by using this individual, I can now modify interactively the values in this panel to explore the dynamics of the model and the effect of the different molecules. So I can start by increasing the value of delta, for example. If you remember, delta is a parameter that drives the activation of cytotoxicity by bevacizumab. So if I increase the value by entering 10 with my keyboard, it increases the effect, so I can see better the effect of bevacizumab. Be careful that I am in log scale here. And next, I can now focus on the treatment panel here to change the doses that are used for the predictions. If I click on each treatment element, I can see the doses that are used for this prediction. And as in the initial data set, 3 doses of each molecule are given. So for bevacizumab, I have these 3 doses that are separated with a 14 days interval. But to simplify the prediction, I can choose to remove, for example, the second and third doses for each treatment. So this is what I will do now, to keep only the first dose for bevacizumab, cisplatin, and pemetrexed. So now I have only 1 dose that is given at day 17 and the effect here. And I can now see what happens when I switch the dosing time of the bevacizumab dose to administer it before chemotherapy, so by changing this dosing time here. To compare with my current prediction, I create a reference and I decrease the day of administration.
57 of the programs that and then.
So by decreasing the day of administration, I see now a stronger effect when I give the dose 1 day before the dose of chemotherapy. So I can create a new reference and decrease again. And I see that if I give bevacizumab 2 days before, the effect is quite similar, but a little bit smaller. So it seems that 1 or 2 days seems to be the optimal gap to have the strongest treatment effect, at least for the typical individual based on these typical parameter values. But is it the same when I simulate the whole population and take into account the variability in the model? This is what I can check in the tab simulation, where here we can simulate whole populations of new simulated individuals. So here I have defined 4 simulation groups based on different treatment schedules. In each case, the schedule is quite similar to the schedule in the initial data set with 3 doses of bevacizumab and chemotherapy. But in the first group, bevacizumab is given concomitantly. So at the same time as chemotherapy. In the second group, it is given 1 day before, then 2 days before, and then 3 days before. And after running this simulation for large populations, I can compare in the resulting plot the differences in tumor size in the 4 groups. And I have added a horizontal line to mark the median tumor size at day 60 in the group for 1 day, between the bevacizumab and chemotherapy, which gives the smallest tumor size at day 60. So this seems to be the gap that gives the best result. Now, to have a more detailed result, I can also take advantage of the possibility of scripting in R the simulations done in Simulx. So this is what I have summarized here. I can repeat the simulation for a large set of different time gaps with small steps between the time gaps to get this figure, which gives a more precise idea of the treatment effect with respect to the gap in dosing schedule. So to obtain this figure, I have used the R package called Lixoft connectors, which contains a list of R functions to call Simulx and to perform all the actions that I can do in the interface, but from R. And this allows me to define a loop that performs a new simulation for each time gap in this sequence. So each time gap on this axis with a step of 0.1 days. And I use some functions from the list of connectors to load the project, to modify the group element to switch the dosing time with the gap, and then run the simulation and get the simulation results. And then I have not written the rest of the scripts, but this would be just some post-processing of the results to get, for example, the mean tumor size at 60 days as an endpoint of the simulation. And with this, I can now find that the optimal gap for the mean tumor size at 60 days is this minimum here. It corresponds to 1.1 days between bevacizumab and chemotherapy. Moreover, I can also notice on this plot that the efficacy loss in scheduling a shorter gap, that in scheduling a greater gap here than optimal is much less than the efficacy loss in scheduling a shorter gap, as I can see from this slope. So this is also an interesting result. So to conclude, I have estimated the population model of tumor dynamics on my whole data set, and I have used this model to simulate, to perform new simulations and to show that a gap of 1.1 days between bevacizumab and chemotherapy gives the smallest tumor size at 60 days. And I have also found this interesting result on the efficacy loss with a greater gap. The next step here would be to extrapolate the model to human, to use this model to predict optimal dosing schedule in human, and this could be used to plan a clinical trial in the next phase, for example. So this finishes my presentation. And now, if we have the time for questions, I will be happy to answer them.
Thank you very much, Pauline, for the excellent and insightful presentation. You ended just about on time. So I think we have time for maybe one question which has come in. Let me go ahead and just take a look at that.
Right.
So Pauline, you show how Simulx conveniently includes a fully documented open source library with many PK/PD models included. And in the example here, we talked about working with a compartmental PK approach to connect to the PD outcomes. How easy would it be to apply simulated PK data coming from, let's say, an external PBPK model and feed that into the Simulx platform to build up the PBPK/PD system?
It's an interesting question. I think depending on the format of the result from the simulated data, it would be quite easy to feed directly the data in Simulx to estimate a model, or to use directly the estimated model in Simulx to combine different models together and get an extended model.
Okay, good. Well, thank you very much again for the presentation.